Good afternoon, guys. My name is Kim.
And I'm Yeshua.
Today's topic is about monohybrid and dihybrid crosses. First off, let's talk about crossing. What exactly is crossing?
Crossing is the breeding of two parents that have different genes but create offspring that share traits of both parents. Wait, what about Gregor Mendel? I remember his name from somewhere.
Gregor Mendel was a monk that dedicated most of his life to science and knowledge. He is known as the father of genetics for his most important discoveries in biology.
Wait, I have my info here, and apparently he discovered how the passing of traits or genetics were inherited, which is a study of heredity. He experimented with pea plants and cross and self-fertilized the plants.
He even came up with something called the law of segregation.
Oh yeah, it states that when an organism creates gametes, each gamete receives just one gene copy that is selected totally randomly.
He also came up with the law of independent assortment. It stated that the alleles of two or more different genes get sorted into gametes independently of one another.
In simpler terms, the allele a gamete receives for one gene does not at all influence the allele received for another gene.
Mendel discovered that each trait is controlled by two factors, genes and alleles. Genes are factors that determine your traits, while alleles are different forms of the same gene.
Our next term is generation. There are three generation categories: F1 generation, F2 generation, and parent generation.
Parent generation is the first organisms to be crossed. The F1 generation is the offspring of the parent generation. The F2 generation is the offspring of the F1 generation.
Well, what about dominant and recessive alleles?
Dominant allele is more visible/noticeable even when the other allele is present. They represent the capitalized letter and are the stronger gene. Recessive alleles are hidden whenever the dominant allele is present. They represent the lowercase letters and are hidden.
Heterozygous are organisms with two different alleles and are dominant traits. Homozygous are with the same allele and can be dominant or recessive.
A homozygous dominant will look like two uppercase letters, while a homozygous recessive will look like two lowercase letters. A heterozygous will look like one uppercase and one lowercase, with one being dominant and one being recessive. Okay, now what in the world is monohybrid crossing? Also, why the heck is the board so dirty? Anyways, monohybrid crossing determines the allele combinations of offspring for one particular gene. Today we're going to compare brown eyes and blue eyes. The scenario for today is, what is the genotype of a brown-eyed person who marries a blue-eyed person, who then produces a blue-eyed offspring? Since the allele for brown eyes are more dominant than blue eyes, we will label it as capital B. On the other hand, since the blue eyes are more hidden, we will label it as lowercase. Now, because the blue eyes are crossed with brown eyes, you should write BBXBB. Now we have to make a Punnett square. First, you write the dominant trait on top, along with the recessive trait on the side. Now let's watch the magic unroll. And there you have it. Half of the progeny are going to be the possibility of brown eyes, while the other half is the possibility of blue eyes.
So now I write down the alleles for each parent dog so I don't get confused. So the capital letters are the dominant and the lowercase are the recessive. H is hair and B is barking because we're comparing the barking and hair traits. Now I draw up my 4x4 table where we're going to cross the traits. And now I'm going to use the FOIL method with F standing for front, right? O for outside, I for inside, and L for last for each position. So you see me doing that to make sure I have all the possible combinations of gametes. Now I'm going to do the same thing for the second dog on top of the table. Now I'm going to fill in all the possible combinations so we can make our prediction. I'm really taking my time, as you can see. so as not to make any mistake, so we don't confuse our results. And I made a slight mistake right there, but I fix it soon. Yeah, right here. And I made another mistake, but meh. I'm outlining each column because they're all the same, and that's going to be easier to organize. So I have a hairless dog that doesn't bark, hairless dog that barks, hairless hairy dog that doesn't bark, and a hairy dog that barks. So we have 4 of each. That's 25% for each, or 1/4 for each, or a ratio of 1 to 1 to 1. And that ratio is for genotype and phenotype. This was Kim
And Yeshua.
on KNY Science Show, teaching you the basics of genetics.
See you next time when we teach you on whatever we teach you.
Bye.
Bye. Now I can go to sleep. Oh my god.